Right, thank you very much, Creighton, for this nice introduction. Thanks everybody for coming to my presentation, and I hope I will succeed in give you some idea about the work we are doing, about the importance of using mathematical models to understand metabolism and to make a basic foundation of knowledge to make metabolism more useful for us humans. The outline of my talk, I like first to start with an introduction, basically giving some overview about metabolic networks, about metabolic flux, and also about the organisms we are using in our research and what's the main economic interest behind those organisms. Then in two parts, first I focus on how you can probe metabolism or metabolic fluxes with a more experimental approach of probing storage metabolism. So this is the part based on isotope tracers, flux analysis based on isotope tracers. And then in a second part, flux analysis and analysis of metabolic networks without isotopic tracers, more as a kind of in silico type of analysis. At the very end, I like to give you some insights into ongoing and future research. I like also to mention that I try to get the right balance in this talk. Of course, it can't be really detailed mathematical issues. So, but I also hope at the same time that this talk gives you a good insight into this work. So, in the near future, I think most of us would agree that renewable material from plants is very important for things like biofuels or bio-derived materials, at least in part, replace our dependence on fossil fuels. And within the Department of Energy, the Office of Science, the section of Basic Energy Sciences, one of their basic mission statements includes the provision of foundational knowledge on biochemical processes that take place to convert sunlight into storage and store sunlight in storage compounds, sunlight and carbon in storage compounds that plants and other organisms can make. So insofar, I think that my interests also fit very well in into this framework. And now, let me start by saying we would like to design a plant to fit our needs to store energy and carbon. And plants very often store energy and carbon in seeds. So during seed development, CO2 that is fixed primarily by photosynthesis is transported into those structures like a seed pod, and there it is used to synthesize storage compounds like starch, protein, and oil. So we would like to be able to change this seed composition, make more oil, less starch, something like that, or to introduce a different storage product and let seeds store something different. Or in recent time also there are many research efforts in going to use the storage mechanisms that you find in a seed and put them into leaves and other parts of a plant to increase the overall storage capacity, for example, to store oils. And the obstacles to be overcome if you try something like this are all lying in the inherent complexity of metabolism and the resilience of metabolism to just easily manipulate it. And so my talk hopefully will bring this idea close to you. So I will start to give some introduction about metabolic networks, metabolic flux. So if you look at this map that shows the traffic of trucks on national highways, and thickness means how much traffic is going on. So you see that between East Coast and West Coast, the traffic distributes in a certain way between different routes. And so you see it's a complicated network, and it is a continual flow, more or less. And so that's a good analogy to what's going on in a metabolic network in an organism. Another analogy would be this. You see here the production of the Ford Model T. And so you basically would have here two metabolic pathways converging. There's one from the second floor. So the undercarriage comes out here and gets combined with the top. So this is basically the idea of two metabolic pathways merging. And you could think of the workers in these assembly lines at each stage. That would be the enzymes. At each stage, you have a specific task of modification, and your metabolite moves on. And here at two merging pathways, you can also make clear one important principle, which is the principle of mass conservation. You always have, you can formulate this in this equation: one undercarriage plus one top equals one car. You can't combine it any different, and that's exactly what enzymes do, or the enzymes are the catalysts of these reactions. So if we can look up something like this in the internet. This is the CAG database, and this shows you the network of metabolism in Arabidopsis thaliana, a model plant. And each dot would be one intermediate, and each connection would be one step, one enzyme step that converts it to the next step. So it shows you an idea about the complexity, and we can zoom out one part here out of the middle. This is something some of you might know from textbooks about basic biochemistry. The glycolysis, this is some part pretty much all organisms have glycolysis that degrades glucose. And we have the citric acid cycle, that's also something all organisms have, and it's also part of this plant. But plants are multicellular organisms, so everything is much more complicated. You might have a leaf that has a distinct function. Within a leaf, you have distinct cell types. These different cell types might have different metabolic functions. Within one cell, again, you have different compartments that have specialized functions, like the plastids have function in photosynthesis, and they have their own, if you like, metabolic network that is connected to the cytosol, and this again to the mitochondria, which are the energy producing, the energy production organelle. So the plant is very complex, and the main focus, of the research I like to do is really to just try to focus on the cell and understand on this level how things work. So we try to get prospective models and experimental models to be able to study this. So during a number of years, we made models of increasing complexity, as Creighton already indicated, and also of different principles of how they operate. And during the years, we published a number of papers that always focus on specific metabolic functions we studied. And but also the interest is to increase the complexity and to give more details into the model. A model is always a very crude simplification of the reality, but we try to improve it continuously. And maybe that might be also one thing we might look into. but I'm not sure about this yet. So I'd also like to give another kind of basic idea about what metabolic flux is. We can look at what's going on in every cell as this hierarchy of flux of information from DNA that stores the gene information, transcripts into RNA, and the RNA gets translated into proteins. The proteins, some of the proteins are the enzymes that are the catalysts of the biochemical reactions. And one way to define metabolic flux is to look at the time scale where things happen. So these things like proteins usually get turned over, means resynthesized and degraded in the order of days. And these small molecule intermediates get turned over much faster. Some of them are totally the internal pool of a cell of, for example, an ATP is an energy metabolite, gets turned over within seconds in the cell. So there's a continuous flow of things going on, and this also gives us the idea of what it means that a metabolic pathway is at steady state, because if there's a continuous flow from this metabolite to that, to the next one, the rates the rates from here and this rate must be exactly the same. There must be some kind of steady state. So that's also a basic idea, an important basic concept about metabolic flux. So this is what we try to study. I now pick up this little example as a minimal kind of metabolic network of a cell. It would be basically an uptake of a metabolite and two parallel pathways and an end product that gets secreted. So we don't know by simply knowing the enzymes and the metabolites, the intermediary metabolites, we simply don't know what's the flux distribution, whether most of the flux goes this way or by this other parallel pathway. So we need a way to find that out, and there is an experimental approach, which is we're using labeled substrate. We're using usually always the carbon-13 stable isotope, which is 1 mass heavier than the carbon-12, and is also visible in NMR. So we would have here a 3-carbon precursor, and we would get it synthesized. Specifically, the first 2 carbons get used with carbon-13, and the third one is unlabeled. So we can now imagine that our 2 parallel pathways in the cell, they differ in their details, but they converge at the same metabolite at the end. So this way, the carbon-1 in some way gets this carbon bond here, it gets broken, and we get something maybe like CO2 that escapes the cell. In this pathway, we get rid of carbon-3 in a different mechanism. So this means these different biochemical mechanisms that, dependent on the pathway that is followed, the label is different. So we can basically just analyze the end product as it comes out, and basically, like counting here, it's 8 of those and 4 of the double labeled. This gives us the flux ratio of this flux versus this flux. This is the basic principle on which all of this kind of carbon-13 basic C13 metabolic flux analysis is based. And this is what we establish in a much larger scale. I give you some more details in a minute about this, but in between I like to switch gears and ask, okay, that's what is the organism we do the research with and that interests us. So we are very much interested in oilseeds because seed oil is a very important product for human nutrition, but also as an industrial feedstock and for biofuels. And there is Brassica napus or rapeseed, which is the 3rd largest source of vegetable oil in the world, but also there are many biochemical studies that detail the biochemistry of the developing seed. Then there is this other plant, Arabidopsis thaliana, which is actually a very close relative. And it is the most popular model organism in plant science, a completely sequenced genome. It has about 27,000 genes. That's more than we humans have, actually, or 20,007 predicted protein encoding genes. So those are very similar, and results are pretty much transferable. If you learn something here, you can pretty much transfer it to this plant and the other way. So I show you some results referring to this and some referring to that plant. And so coming back, give you some details on seed development. So we want to know how this plant stores its carbon resources in a seed, and that's happening during seed development. So after flowering, during about 35 days, there is in this seed, inside the seed coat, something we call the embryo. That's a non-differentiated plant with a little root and 2 leaves. And it grows during that time and finally fills up all of the space. So the seed that we harvest is basically an embryo. So we study embryo development and deposition of storage compounds in this embryo, in the cells of this embryo. And it's actually, the cells are pretty much a very homogeneous kind of cell type, which is basically a large storage cell. This storage cell deposits these oil droplets, and in these vacuoles, large vacuoles, it deposits storage protein as 2 major products. And during the expansion of the cells, more and more gets deposited inside these cells. So we are interested very much in the oil as the most valuable product. If we look at rapeseed as one oil seed with another oil seeds, it's interesting to note that although different species have a wide variety on the content, on the percentage of oil that they can store, within this species, rapeseed, we have very little variation. Also, breeding always tries to increase oil content, but it seems to be very hard to manipulate this. So there seems to be some kind inherent resistance to change this storage composition in the seeds. In order to really study this in detail, we need to take this embryo out of a developing pod, out of the seed, into a liquid culture medium that has all the goodies that usually are coming from the mother plant, and also nice constant growth conditions. During about 10 days, we grow them in culture, and we can feed a carbon-13 labeled precursor and make this metabolic flux analysis. Here, very quickly, a little bit more detail. So we have here this, as I just showed, this incubation, this growth medium, and after the organism, the embryos grow, we extract different metabolites, break them down into smaller parts, analyze them with chemical analytics like NMR or MS, mass spectrometry. This is the experimental part. Then there is here a modeling part, a computational part, which includes the construction of a model that represents what's going on in here, which includes a kind of cyclic process that we can say it's a kind of search algorithm that tries to simulate those labeling signatures and to find the closest match between synthetic computer simulated labeling signatures and the measured signals. It's a kind of search algorithm that finds us finally a best flux estimate. So basically we have typically something like we measure 30 different metabolites with their labeling signatures. These get fed into a into a program. This computational transformation gives us something that maps numbers in terms of fluxes to this kind of metabolic map. So now I'm coming to some results of this kind of study. So if we do this kind of approach with developing embryos of Arabidopsis, which is this model plant, the small plant with complete genome, this is some kind of typical outcome. And I just want to give you an idea. So we have here sucrose as the main carbon source. This is going from here to here is basically what we call glycolysis, which is the same process also other organisms have. And then most of the carbon goes here into the biosynthetic steps of fatty acid synthesis and is transported into, finally, the fatty acids are bound to oil. So we have here some parallelism because of the subcellular compartments like the chloroplast. This is the reactions in the chloroplasts that are in parallel to the cytosol. We have here some of the arrows are shaded in gray. This means that our modeling procedure gives us relatively large statistical uncertainty, which says this is not too certain, but the other fluxes are much more certain and trustworthy. So one example for an outcome out of this kind of analysis is this thick arrow, which is this ribulose bisphosphate carboxylase involvement in breakdown of sugars into lipids. So this is an unexpected result, and we often see other unexpected things if we do this kind of flux analysis. And I will show you some more examples. So now the next, I want to ask the question, how can we, if we are interested in what controls oil deposition, how can we find out what's important? Are the important parts? And biologists really have some pretty crude approaches to do this, if you think what it really is. Because a good analogy is if you ask what are the components required for a good sound in a radio, this actually goes back to this whole example to a paper published in 2002. So basically, the biologists would make a kind of rip-out approach. We rip out some parts randomly or systematically, one after another. We need thousands of radios to do this. And then characterize it. Does it work or not? Or how is the sound? And this is a basic approach. We first go and apply it to organisms like a plant. So we have to go through 27,000 genes. So we produce thousands of mutations. This is actually an easy part because you easily can mutagenize plants. But then we select for particular phenotypes like a wrinkled kind of seed, which is typically if the seed can't make any oil. So this is basically an approach that has been done by in particular, this approach for oil synthesis has been made by the group of Christoph Benning years ago, and they isolated a mutant that really makes much less oil. So if we grow the embryos in culture, we find that they make less oil than the wild type, and this is compensated by somehow making more protein and also free sugars. So we now want to use flux analysis to study to see in more detail what's going on. So this is again the wild type flux distribution, and if we compare it with this mutant, we see that all of the carbon flows towards fatty acids and oil synthesis are largely reduced. We also see some other effects. So first of all, I have to tell you these circles, those denote where we have knowledge in the meantime that these enzyme steps are reduced in this mutant. And this is because this so-called wrinkled 1 mutant is a transcription factor that controls the expression of multiple steps along this pathway. And this is very seed-specific. And we see really that the flux is largely reduced. That's what we expect. But we also see some kind of unexpected things like we can look at this as a bypass from this metabolite in the cytosol can be then transported here into the plastid. So this kind of thing also happens that this bypass is activated and can compensate at least in some part for the loss of enzyme activity here. We also have here some increased flux in the citric acid cycle, which is not really expected. So we see some kind of side effects or some kind of unexpected effects that are of interest. A different way to ask what makes a high oil seed versus a lower oil seed is also we just look for different genotypes that differ in their oil content, and then we use it in our embryo culture method and make the flux analysis. So what we did was, in collaboration with Hardy Roletschek in Germany, he had access to a large seed collection of different varieties of rapeseed, and so he screened for a number of genotypes that we then further studied in detail with flux analysis. So we have here 11 genotypes that have a variation in oil content from about 25 to 40%. And we made, for all of them, we made basically the same flux analysis approach and producing the full flux map, so to say. We also make in parallel things like enzyme analysis, metabolite profiling, proteomics, and other omics approaches, but I here like just to focus on this part. And this is again the flux modeling. I just want to show you, to give you an impression how much data here color-coded, the labeling signature data that go into the modeling process, 11 genotypes. And so also to give you an idea of the scale, computational scale, to make all of the things we need to do to produce the flux maps. We used about 1,000 CPU days, so that was good that we have access to the Brookhaven Linux cluster. So here is one of the genotypes, the flux map we obtained, and it actually, you might remember, it looks pretty similar to what you see in Arabidopsis. So things are pretty similar, in particular with this, in this part here. I'd like to focus a little bit on this region. Again, I discussed before this kind of two parallel reactions, and also this branch, I like to add this branch, and to make some interpretation of the results of these 11 genotypes. So here it is simplified. We basically have here the flow of carbon from glucose to this phosphoenol pyruvate intermediate via pyruvate to fatty acids into oil. We actually have 3 alternative routes it can go, and you saw them in the former flux maps, and you saw how much carbon is flowing. Now we basically can make a correlation across 11 genotypes and ask if correlate oil content with flux. And what we can see pretty clearly here is that this route basically is not responsive. It's not increasing with increasing oil. This one also not. It's only this one, which is the plastidial reaction, the plastidial pyruvate kinase reaction, and those reactions. Those are upregulated. The flux increases, which means they must be upregulated for an increase in oil content. This means what we saw before, that exactly those reactions are also controlled by the wrinkled transcription factor gene. So this fits the idea that wrinkled is a very important regulator in seed oil that controls seed oil. So now I want to move on into more theoretical in silico prediction of storage compound synthesis. So we don't use in this approach these carbon-13 tracer techniques, but we are able to use a much more detailed model, which we actually can't use in with carbon-13 tracers. So this is a network that we published last year that has 572 reactions, and it represents in all detail many different subcellular compartments and their connections. So this means these are different pathways that contain a number of reactions. So this compartment has 206 reactions, for example. This means we have 52 transporters here connecting those two compartments. So it's a quite increased complexity and more realistic in this sense to the real metabolism. The model has basically multiple substrate uptakes. It has all of these parts of metabolism, and it has synthesis parts for the synthesis of all of these storage compounds. If we pick out glycolysis, for example, again, you saw this before, it would be this way that we formulate glycolysis. If you remember the assembly line where I said it's simple stoichiometry, and again, all of the reactions are formulated by their stoichiometry, by things that are combined, substrates to products. So how can we use this model to predict flux distributions? So I don't want to go into much detail, but what we basically have as an input to the model is we derive biomass synthesis rates and fluxes into biomass, so those here, based on the biomass composition that we can measure by extracting and analyzing those embryos or seeds. But we don't use CAM13 tracers. Instead, we use an optimization procedure that is called linear programming that helps us to predict flux distributions by saying we want to search for flux distributions that have minimal substrate uptakes. This makes sense because we assume that the whole process has a big degree of optimality. You don't want to waste the substrates like sucrose that the embryo receives from the mother plant. So optimality is a good criterion, minimal uptake of substrates. And based on this, we predict fluxes for all 572 reactions. What we can do now with the model, in difference to the experimental flux analysis, is we can change this composition at will as a kind of predetermined outcome. We can say, we want the seed composition be in a different way, and then predict fluxes and ask how is in each of these pathways the fluxes predicted to be different. So we focus here on those two fractions of protein and oil, or this tag means oil, and ask predicted the fluxes in the metabolic network dependent on a trade-off of those two components. Like in the one extreme, we have 51% oil and no protein, and this trade-off is there's a linear trade-off into the other extreme. So we can simulate across this trade-off axis the flux distributions. We have some reactions that would go down like this, and some that would go up. In this case, we would say this reaction it reacts in an oil-responsive fashion, because if we increase, if the flux or the absolute value of the flux increases with percent oil, it means that the flux through the reaction has to increase. The other way, it would be protein responsive, because thinking the other way around, it increases with the protein content. So we basically categorize all reactions in the network now quantitatively. We basically take this slope, and we reference it here to what I will show you only. We reference it to this biomass across the trade-off now, and we calculate the slope, the slope in this the linear slope, and sort and rank the reactions by their slope. So and then we simply ask which are the top 10 ranks in slope. So which are the top 10 most sensitive reactions to oil increase, And we find here mostly reactions of glycolysis. And if we ask which are the top 10 ranks of protein responsiveness, we find, interestingly and a bit unexpectedly, that these are mostly reactions of energy metabolism. These are reactions in the mitochondria that are related to the production of ATP, which is the most important energy molecule, and delivers energy for many biosynthetic reactions in the network. So this was pretty clear. This result is pretty clear because I showed you before that apparently many reactions of glycolysis are regulated by the wrinkled transcription factor. So this makes sense. The most sensitive reactions are regulated by this transcription factor. This makes, again, this is a little bit anticipated, and we are very interested to follow up basically this lead in more detail. So then also one particular reaction here in this ranking is acetyl-CoA carboxylase. This is considered a key step in fatty acid synthesis, meaning it is basically the product of this enzyme goes only into fatty acids, nothing else. So the idea people before had is if you upregulate, if you give more enzyme of this reaction, then you might have more flux and more oil production. So we can compare here a former experiment that has been done with the model in a certain way. We can ask, in a study where acetyl-CoA carboxylase was overexpressed in rapeseed, they found an increase in oil of about 10%, and they also measured the enzyme activity in those seeds, and they found that it was increased by about 8-fold. So you have 8-fold more, apparently 8-fold more enzyme. Now we can basically transfer this increase into the model and ask if we have an increase in the model by 10% in oil content, what is the expected increase in rate of this enzyme, of this reaction, of this enzyme step? So, the outcome is that actually the model only needs a 1.3-fold increase in flux to accomplish a 10% increase in oil content. So this would mean that theoretically we might not need an 8-fold increase in enzyme. So this seems to be like not fully effectively used. They put in much more enzyme than they got an increase in flux. So one explanation for this comes from a basic insight in metabolic control theory, which is the limited control of enzymes over pathway flux. So I'd just like to visualize this concept. If this is a wild type pathway, this is the pool size, the concentration of a metabolite, and this is the next metabolite. This is the concentration of an enzyme, and these are other enzymes. If we increase this by 8-fold, and we have only an increase in pathway flux by 1.3-fold, what might be the explanation? One explanation might be that in this steady state, we might have a reduced concentration of this intermediate. Now just think about the assembly line where you have different stages and you would decide to give this stage just 8 workers instead of 1. From this it is clear that you don't increase the flux by 8-fold because maybe this step is just not able to deliver in the same rate than this step can theoretically use it. So there will be a steady state will be found that has here a reduced pool size of this intermediate. Another explanation, in particular, that applies to this acetyl-CoA carboxylase is that we know that this enzyme is inhibited by the product. So if some of the following intermediates accumulate because they can't be processed fast enough into oil, they inhibit the enzyme. And this is actually one thing that just recently was in the group of John Shanklin. They are working on this particular enzyme, and they just 2 weeks ago published one of those inhibition processes in this enzyme in PNAS. So as a summary, let me quickly summarize. So we are interested in the functioning of metabolic networks, and in particular, we want to know what determines oil and protein synthesis in seeds like Arabidopsis and rapeseed. So we use two different approaches. One is the experimental flux analysis approach, where we use carbon-13 tracers, and we can see something about the in vivo pathway usage, and we might see some unusual or from the textbook coming from the textbook unanticipated usage of reactions and pathways. We can compare different genotypes, mutations with the wild type, and see the effects on flux redistribution. We can correlate different genotypes. If we have a series of genotypes, we can make flux correlations with our interesting traits, the oil content. All of this helps us to learn more about the system. The predictive part, we can predict flux values by this pre-specified biomass composition. We can use that to find candidate genes that can be experimentally explored to increase oil. And we can make a quantitative assessment on the effectiveness of a certain genetic transformation. With all of this, we have the tools in place to make designs of metabolism, and to verify, in particular with this part, to verify the outcome of these attempts of modification. So now a quick idea about current and future work. I showed to you this simplified scheme before of these alternative pathway leading to the same product, and accordingly, we currently working on transgenics that overexpress this enzyme or that overexpress wrinkle, the transcription factor that regulates all of this. So we are working on those transgenics and then like to analyze them with our C13 flux analysis. We also have from our collaborations with Bayer Crop Science about 10 transgenics in the work. Transgenics in central metabolism that we will further analyze. We also try to expand our work to use other organisms that are, on the one hand, relevant as bioenergy crops, and on the other hand, also useful as good laboratory organisms. The idea is, for example, if we learn something here, if something is effective here, we can try it in the other organism or in this organism. So this is duckweed, which is a very fast-growing aquatic plant that floats on surface of ponds. And now in the summer, you see sometimes ponds totally green with this green surface. It grows very fast, it's very, very productive, and we might be able to modify it to make oils. So we do this in collaborations we established with the group of John Shanklin and Shangsheng Su. We also work with in collaborations on microalgae, which are also aquatic organisms that grow very fast and are potentially producing, so those can produce oil in high amounts. So we try to have all of the methods in place for genetic transformation. We have a genome sequencing project for duckweed, one with JGI and one with Cold Spring Harbor in collaboration. We also work on RNA-seq, on sequencing of transcript messages to being able to better determine the enzyme set in each of those organisms to reconstruct the networks. So this is one of the basic concepts we want to go and expand in the future. Now I'm coming to acknowledgements. So we have our funding from the Department of Energy, Basic Energy Sciences, and also recently from EERE. We got also currently still an LDRD on establishing one new model organism from Brookhaven Lab. We have funding from Bayer Crop Science on increasing rapeseed oil yield. So some of the work indicated this, I showed you. We got, I thanked Christoph Benning and Sebastian Baut for obtaining some mutants, where I showed you some data about it. And I also like to thank my current co-workers in the lab who are working on different projects. Jordan Hayes doing all of this computational work on different modeling approaches I didn't tell you about. Hai Shi is working on flux analysis and modeling of the duckweed. Xie Sun is working on the microalgae, on flux analysis and modeling of microalgae. Inga Hebelmann is our person who knows how to genetically transform organisms, and she also is working with the flux analysis of the rapeseed. We have collaborations also here at BNL. I like to acknowledge with Alistair Rogers, who has an enzyme profiling platform where we can basically analyze our materials and quantify enzymes of interest. Also our German collaborators that measure metabolites in our samples that also are involved in the Bayer project. I also like to thank those colleagues and more colleagues, but in particular those colleagues, John Shanklin and Bill Studia, who gave me in the last years lots of advice on different issues in establishing a lab and how to run a lab and these things, and collaborations with Shangxing Liu and CJ, we always say CJ Liu, Shangxing Su, and Alistair Rogers. Thank you very much for your attention. So if you have any questions.
Okay, we have time for questions. Okay, if you solve the problem of finding, say, the best country with the best oil, one problem is that it takes up a lot of real estate, and so my question is, have you guys thought of, like, say, ocean plants to, like, seaweed or something like that, which you could oils from that, since you don't have to grow on land?
Oh, yeah, so if I understand it right, you refer to the amount of land you need, to grow a certain bioenergy crop. Well, one first remark I like to do to this question, not really an answer, is we are really much interested in the basic science of functional metabolism, but of course you need to have a perspective about how is this usable. And one thing I can say about this that there's in the EERE program, there's lots of research going on in assessing resources for aquatic organisms, for example, for duckweed or for algae, and making calculations about what's the capacity to produce these at large scale. But you are certainly right if you would estimate that the resources of agricultural area to produce biofuels are somehow limited. But to my understanding, for these programs on bioenergy in particular, I would say that it's certainly not the idea to have one solution. It's the idea of have a portfolio of different solutions. So bioenergy crops should have a contribution in the future, but no one says it's all bioenergy or biofuels. So that's my idea on this. Yes.
But is it also not true that if you're able to make the plants more efficient, you get a lot more product off the same amount of land? And so that's another.
That's yes, sure. So that was kind of a question, or a comment from John, that of course, the more efficient we can make the productivity of those crops, the more we can get out from per area. That's clear. Yes. Yes. So the question was how basically, I guess, how much material we need to make this stable isotope labeling experiments. In plants, we need something like, we can make one experiment by using one plant. So we have those, I showed you these pictures with plants having these pods, seed pods. We need like only 5 seed pods or something like this to make basically one experiment. In case of Arabidopsis, which has much smaller seeds, is actually more tricky. But so the requirements of material are not too big. It's maybe the requirements of growth chambers to grow enough plants is a little bit a problem. So if we want to go to other organisms like animal cell cultures, I think, I don't have a good idea how much cell material you can get out of one culture flask if you make those mammalian cell cultures or something like that. But an idea about, from the plant material, you use something like 50 to 100 milligram fresh weight. So I don't know how much cell culture you need, yes.
How do you know when the model networks are big enough or good enough? You haven't left out some species or some enzymes or some inhibition pathways that might change the whole story?
This is of course this is of course always a possible limitation of the validity of these studies, and we are constantly trying to. I could present you from the work we have done examples where we even could find, based on labeling signatures, that we are missing something in the model, and then we looked about in literature or we looked with enzyme assays about possible enzyme activities and found them and can then add them to the model. One important approach to mention is, of course, that with the advent of more and more completely sequenced genomes, it is also possible to go directly from a genome to identify each possible enzyme gene product and transfer this into a huge network. But this has also some limitations about the knowledge of all of the enzymes.
So yes. I'm going to ask one question, and it's probably not a surprise to many, but when will you be able to help us with symbiotic relationships between plants and microbes, such as in nitrogen fixation?
Well,
is that a stretch goal or is that an achievable goal?
Well, it depends on the. So, if I'm thinking about clearly the experimental approach, you need something where you have the symbiosis that in some way you can keep it in a steady state or approximate a steady state during your experiment. So that's one clear requirement. This is a requirement that often limits actually the possibilities about what kind of systems we can study. Many things are not easily growing in a kind of steady state.
How do you actually detect carbon-13?
Carbon-13 is one thing. It's a heavier isotope than carbon-12. So in mass spectrometry, we find isotope peaks. and we typically fragment molecules. So the goal is typically have a molecule with multiple carbons fragmented into different fragments that have only parts of the whole molecule, and then if we see isotope peaks in the subfragments, we get most of the labeling information out of. And then the other possibility is using NMR, so we can measure carbon-13 enrichment in NMR spectra. Yes.
One last question. So excellent talk. I have to ask a
Thanks.
question. So your whole modeling approach is underpinned by the assumption that all life processes are in steady state delivery. And yet, you showed an excellent slide which suggests that life is not in steady state. It's not a steady state environment. So for example, product inhibiting upstream enzyme function are likely precursors to that pathway to provide positive feedback downstream. So how do you account for the non-steady state process?
So the question was about the steady state assumptions we always have in the model, in the carbon-13 flux analysis, or in all the modeling we are doing, and how this is realized. And I showed one slide. If you remember one of the slides, I showed where you have different time scales on which things are happening, and clearly, usually an organism, if you have time scale of days, you should expect that metabolism is not steady because gene expression and physiological environment change, and it has constantly to adapt. So we always try to use an experimental setting where we can at least approximate during several days that all of the physiological conditions stay constant, and we basically approximate a constantly growing cell. And the gold standard, so to say, for this kind of analysis has been established in microbial engineering, where they have microorganisms in a completely controlled, called chemostat, where everything, all nutrients, concentrations, pH, temperature, everything is kept constant, and they constantly multiply the cells at a constant rate. And I have to say that even at this, for this, so to say, gold standard, there are also deviations from steady state known, but we have basically to take the best approximation, the best experimental closeness in order to derive those flux maps. But I would agree certainly, that in real life things are often in transition. Yes.
So let's thank Jörg one more time, and there'll be refreshments outside.
Thanks.